In this video, we are going to discuss section 4.6 on cellular structures, and here are our learning objectives. We'll start off with our membrane-bound organelles. Remember, these membrane-bound organelles are going to be surrounded by a membrane, and we will see that they'll allow activities in an isolated environment. So we'll start off with our endoplasmic reticulum. This is an extensive interconnected membrane network. We see that it varies in shape, but it is one continuous lumen. And it is extending out from our nuclear envelope to the area of our plasma membrane, which we don't see pictured here. It's composed about half of membrane within the cell, and the point of attachment for ribosomes would be within our rough ER, and that's why we call it rough ER, because it has these ribosomes attached to it. Whereas when we have our endoplasmic reticulum without the ribosomes, this is called smooth ER. And you could see in this photo here, this is a electron microscope mictograph, and these spots here are representing the ribosomes, whereas these channels or membranes are representing the actual rough ER. And here's that figure again for you. Now, our rough ER is involved in protein production with those ribosomes that are attached to it. The original structure of a protein is going to be changed here and then transported out in the enclosed membrane sacs. So that's what we see here, where the transport vesicle can be found. It shuttles proteins from the rough ER to the Golgi apparatus that we'll talk about in just a moment. Our smooth ER is involved in diverse metabolic processes that vary depending on the cell, but its overall function is to synthesize, transport, and store lipids. We also have carbohydrate metabolism associated with it and detoxification of drugs and poisons. So the main thing, in order to differentiate these, with rough ER, I always think proteins. With smooth ER, I think fats and carbs and detox. Now let's talk about that Golgi apparatus that I mentioned earlier. It is composed of what we call cisternae. They're these elongated sac-like membranous structures, and we're going to see that it exhibits polarity. On this side, we call it cis face, which is more proximal to the ER, so we see that over here. So this is the cis face. And then on the opposite side, we call this trans face, meaning it's further away or distal from the ER. Its main job is to modify, package, and sort the proteins. So you can kind of think of this as like your postal office. It tells you if your package is not done correctly, it can repackage it, and it sorts it into the specific area. We'll also see that it forms secretory vesicles, and these are going to become part of the plasma membrane. Others will release contents out of the cell, and some of them may even fuse with lysosomes. And we'll talk about what lysosomes do in just a moment. And here is a photo of a Golgi apparatus in a mictograph. And here is our lysosome. These are going to be small membranous sacs, and inside of them they contain digestive enzymes formed by the Golgi. So they're going to assist to digest any unneeded substances. When we talked about phagocytosis in our previous sections, it is cell eating, right? And the way that phagocytosis takes place is one of the vesicles that contains that solid particle within it will fuse with a lysosome, and the digestive enzymes in it will help to get rid of or break down whatever substance is in that vesicle. Next, we have peroxisomes, and a lot of people will mix up peroxisome with lysosome. I mean, they look similar, right? So notice how this lysosome in a mictograph is darker versus this lighter view of the peroxisome. This too is going to be a membrane enclosed sac, but it's going to be smaller than a lysosome, and it's pinched off vesicles from the rough ER. Our proteins will be incorporated to serve as their enzymes, and the metabolic functions here are going to include a role in chemical digestion, beta oxidation, and lipid synthesis. And now we'll discuss the endomembrane system. Essentially, this is going to include our ER and our Golgi apparatus, the vesicles that we discussed, our lysosomes and peroxisomes. So using these membrane-bound structures, we are going to see that we can make a connection directly or through vesicles moving between them. And this provides a means of transporting substances within the cell. The rough ER makes the proteins, and then we're sending it to the Golgi apparatus for it to be modified, packaged, and set up in storage. Now let's talk about mitochondria. Note that mitochondria is the plural form. Mitochondrion is the singular form. So we see an oblong-shaped organelle here, and it has a double membrane. This organelle is really important in aerobic cellular respiration. It's going to give us the most ATP. So a lot of people call the mitochondria the powerhouse. Now let's move on to our non-membrane bound organelles, starting with ribosomes. Our ribosome is going to contain protein and ribonucleic acid and is arranged into a large subunit and a small subunit that come together. And the large subunit has these special sites, which we're going to talk about in our coming sections. We've got our E, our P, and our A site. This is made within that nucleolus, within the nucleus, and is assembled into its large and small subunits coming together within the cytoplasm. Remember, we talked about our rough ER and how we had ribosomes bound there. They were going to synthesize proteins for export or to become a part of the plasma membrane or serve as enzymes in lysosomes. And we also had some free ribosomes just sitting within the cytosol, and all other proteins will be synthesized there. Now let's talk about centrosomes. So our centrosome are these two components together. If we are talking about just one of them, then we would call it a centriole. And it's surrounded by an amorphous protein. Its primary function is to organize microtubules within the cytoskeleton. And we also see it doing this when we go through the process of mitosis. So that's coming up in a section to see a visual of that. And now on to our proteasomes. These are going to be large barrel-shaped protein complexes. They are protein digesting organelles, which the protein portion of that should remind you of the word protein, so you can remember that it is going to break up those protein molecules. And we're just going to find this within the cytosol and the cell nucleus. We're degrading cell proteins through an ATP-dependent pathway. So any damaged proteins or proteins that were folded incorrectly or proteins that we just don't need anymore can all be degraded. And our proteins marked with ubiquitin tag for disposal will have these proteasomes coming after them. With age, we may be unable to normally remove these damaged proteins, so they tend to linger along longer than they should. Now let's talk about the cytoskeleton. This is going to play a role in our intracellular support, organizing our organelles, cell division, and movement of materials. You could see that this is just a portion of our cell over here, but the cytoskeleton extends throughout all of this region. So what we see is that 3 proteins are used in order to create the cytoskeleton. We have our microfilaments, our intermediate filaments, and our microtubules. Our microfilaments are the smallest components of our cytoskeleton. Our actin protein monomers are used, and they are 2 twisted filaments. So you can kind of see the individual subunits here in green, and they are, imagine 2 long strands that are just twisted amongst one another. They're going to function in maintaining our cell shape and internal support and cell division. Our intermediate filament is going to be, surprise, intermediate sized for more rigid than rigidity than our microfilaments. Their job is to give structural support and help with cell junctions. Lastly, we have our microtubules. They're going to be the largest of the cytoskeleton and composed of a protein known as tubulin. They may be elongated or shortened as we need them, and their job is to maintain the shape, help with cell transport, and cell division. Now let's look at some of our cell's external surface structures. First, we have cilia. These are the hair-like projections that are going to move substances along the cell surface. For example, in our respiratory passages, we have cilia that help move debris out of our respiratory system. Flagella is another external surface feature. It is longer than it is wider compared to our cilia, and it helps to propel the entire cell. So the best example that we have in our body or in male bodies is the sperm cell. And then we have microvilli. These are going to be extensions of the plasma membrane, and they really help to increase the surface area. So areas like our small intestines, where we are doing a lot of absorption, we would want to increase surface area so that we can absorb more nutrients into our body. Next, we're going to discuss membrane junctions, meaning areas where cells and cells come together and make contact. So first, we have our tight junction. This is when we have strands or rows of proteins that are going to link the cells together, and it prevents substances from passing between the cells. So this would be really important, like an area of the brain in which we create something called the blood-brain barrier, so certain toxins can't get into our blood supply to our brain. So we require materials to move through rather than between the cells. Now our desmosomes are up next. These are composed of proteins that bind neighboring cells. We also have hemidesmosomes, in which we have half of this structure here, and that helps to anchor the basal layer of cells, meaning the foundation or bottom portion of the cell of the epidermis, to underlying components. And lastly, we have our gap junction. These are going to form tiny fluid-filled tunnels. So you can kind of see this tiny tunnel over here, and they provide a direct passage for substances to travel between 2 different cells. So this would be like cell 1 over here, this would be cell 2 over here, and an ion would pass from cell 1 through the tunnel to cell 2. And this would be really important in the cardiac muscle, where we need those ions to travel in between the muscle cells to cause contraction. And here is just the full figure. I placed just the close-up view of these junctions, but you can see them in place in between the cells here. So here is our tight junction. Here is an example of a desmosome. This is a hemidesmosome, which is showing the one cell attaching, let's say, an epidermal cell to the dermis down here, just for example, but we can have hemidesmosomes in other places too. And then here is an example of the gap junction that depicts of what it would be like to pass ions between 2 different cells.